Assalamu alaikum, beautiful people. Today we will begin with the study of cancer. What is cancer? A general term for more than 100 diseases characterized by abnormal and uncontrolled growth of cell. What cancer is not actually, it is not immortal cells that will die cell lifespan. Typical cells live about 40 generations. Afterwards, cells build up excessive genetic errors that it results in death. Cancer cells live longer, resulting in greater probability of error accumulation. Clear? Cancer terms are of two: benign tumor and malignant tumor. Benign tumor are invasive growths with the potential to become malignant, and they are restricted to no sorry they are non-invasive growth, while in malignant tumor the cancer cells supply to other body parts via the bloodstream and lymph nodes. Mutagens versus carcinogens. Mutagens are any substance that cause alterations in genetic material. Does cancer occur without a mutation? No. Does mutation occur without cancer? Obviously, yes. Types of mutation include point mutation, when a single amino acid when a replacement of nucleotide occurs in an amino acid, as in case of proline, when third amino acid is changed with A with G, then there is no change, no effect, no mutation. And when first amino acid C is changed with G nucleotide, then amino acid will change into proline, from proline to valine, and that will cause a mutation. Types of mutation include frameshift mutation, in which a complete nucleotide is either missing or extra added, as you see in diagram. Mutation that causes effect: deleted nucleotide is added in another nucleotide sequence amino acid, that causes mutation. Types of mutations include chromosomal deletions, thymine dimers with ultraviolet lights, strand breaks with X-rays or ionizing radiations, DNA ducts, carcinogens, oxidized bases, oxidizing agents. Mutagens versus carcinogens. Carcinogens are compounds that induce serious mutations to DNA that can lead to cancer. Mutagen screening aims test the Salmonella typhimurium. Reverse mutation test. Mutant bacteria that required histidine to grow. Mutation is from a single nucleotide. Okay. Expose bacteria to potential mutagen and grow on a plate without media. Mutagens will randomly reverse the original mutation, allowing bacteria to grow without histidine. If bacteria grow, compound is mutagenic. If bacteria does not grow, then the compound is not mutagenic. And following diagram will elaborate you the whole procedure. Common exposure to mutagens and carcinogens: polycyclic aromatic hydrocarbons, burning fossil fuels, soaked wood pilings, nitrosamines, meat, preservatives, saliva. Common carcinogenic exposures: benzopyrene. Results from incomplete combustion of organic compounds found in cigarette smoke, car exhaust, forest fires, and food. Common genotoxic exposures known to cause cancer in animal studies: produced from the cooking process of animal tissue above 180 degrees Celsius. Higher temperature produces more HCA, that is hydrocyclic amines. Flame cooked meat, baked 354 and 160 degree centigrade exterior only. Deep fry 400 degree foreign, 232 degree centigrade. Flame barbecue 800 degree foreign and 426 degree centigrade. HCl formation from high temperature cooking mutation is induced. Commonly genotoxic exposures, acrylamide, found to be genotoxic in vitro tests produced from high temperature cooking of starchy foods such as French fries, potato chips. Common carcinogen exposures, alcohol and smoking. Synergistic effects. Alcohol dissolves fats in membranes. Carcinogens in smoke enters cells more easily, increase damage potential. As shown, bioactivation. Compounds are more toxic after being metabolized, e.g., benzopyrene. Metabolism occurs via kept for 50s in phase 1 and phase 2 reactions. Bioactivation mutagenic tests are carried out by following procedure. Teratogens. Teratogens are compounds that are genotoxic only to developing fetus. Mother is ineffective. Thalidomide, thalidomide used as a sedative to combat nausea in a pregnant woman in 1950s caused massive development defects through genetic toxicity. Molecular cancer progression model is shown. Clinical cancer progression model is shown, and the clinical cancer patterns. Recurrence of cancer often occurs after the growth has been surgically removed. In 70% of the recurrence cases, cells will differentiate to indicate recurrence. In 30% of recurrence cases, cells will appear perfectly normal, then suddenly become cancerous. Molecular patterns in cancer. Cancer can arise from multiple gene mutations for DNA repair, anchorage dependence, growth suppressors, growth promoters, P53 apoptosis gene. Molecular patterns in cancer specific sequence of gene loss are commonly found in cancer. DNA repair comes first. P53 apoptosis gene comes first. Growth suppressor second. Growth promoter second. Anchorage dependence last. Patterns of cancer. Multiple combination of gene loss can all result in cancer. Cancer at the same site in two individuals often have a different molecular basis. No such thing as a universal cure of cancer. Environmental toxicologist initiation generated an neoplastic cells chemical plus DNA caused mutation. DNA repair apoptosis nothing permanent in the genome. Mutation is irreversible. Gene expression a new plastic cell initiation may never develop into cancerous growths. New plastic cells grow possibly after a latent period, slow process, repeated exposures, benign tumors, dysplasia. DNA repair mechanisms are damaged. Cells still appear normal but increased DNA mutations occur. Progression begins with visible tumors, continued alteration of genetic makeup of the cell, invasion to adjacent tissues, metastasis to unrelated organs via blood, lymph, malignant tumors, hyperplasia, cells lost ability to communicate chemically with each other. Biological repair mechanisms are here. How to be repair damage. Excision repair is divided into two major categories: nucleotide excision and repair, and base excision and repair. Here are the excision process. You can study them in detail, while their detail is not compulsory, so you just have a look on them. Repair of a double stranded break. And thank you for watching. Stay motivated for next videos. Allah Hafiz.